Okay, today's lecture is going to be over cellular respiration. So the first slide, this is an overview slide. Let me get this out of the way. And cellular respiration is a catabolic pathway. If you remember, we've talked about this already when we talked about photosynthesis, which was anabolic. So it's a catabolic pathway because energy that is stored is going to be released by breaking down bonds in this complex process called cellular respiration. So we're going to be releasing energy. Okay, so in the equation, jump down here and look at the equation. It says glucose, which is C6H12O6, in the presence of oxygen, is going to be converted into products known as carbon dioxide, water, plus energy. And the energy is ATP plus, of course, a little bit of heat, because with all catabolic processes, we're going to lose a little bit of heat as a result. But this, of course, is our goal, is to create some ATP. This is a process that's used by cells. This is all organisms. It can be used by prokaryotic organisms as well, even though they don't have membrane-bound organelles such as the mitochondria. So in a eukaryotic organism, this process will take place, most of it in the mitochondria. Of course, some will be in the cytosol. And in prokaryotic organisms, it will take place in the cytosol, plus, let me just put it here, prokaryotes will use cytosol plus their cell membrane, because their cell membrane serves as like the pseudo-membrane of the mitochondria, the cristae. They can use that cell membrane for that. The overall equation, like we said, is right here. This is a redox reaction. Okay, we talked about how redox reactions, one thing is going to be reduced and one thing is going to be oxidized. So in the equation, the glucose, which by the way could be any carbohydrate, this could also be a lipid, this could also be a protein, because this equation, cellular respiration, producing energy, does not have to just use glucose. We use glucose because it's simple and that's what the textbook follows, but you could also use a, like I said, a lipid or a protein to go through this process as well. But for this equation, we are using glucose. That's going to become oxidized, and by being oxidized, it acts as the reducing agent. Okay, it acts as that reducing agent. I have a little hard time with my Mobi right now, but just bear with me. Okay, so it acts as the reducing agent as it's becoming oxidized into carbon dioxide. Now, the oxygen, in this case, is becoming reduced, okay, which means it is acting as the oxidizing agent. And oxygen is one of the most versatile, most common, prevalent oxidizing agents that we have, but it is going to become reduced as it receives electrons and is converted into water. Okay, so that's definitely the job of oxygen, is to receive electrons in this scenario, which is why it's becoming reduced, because if you receive electrons, you're receiving those negative charges, and it is converted into our water. Okay? All right, now looking over here, let me see if I can get this moved up a little bit. Okay, you can see the steps that we're going to go through in cellular respiration. Okay, the big name is cellular respiration, and this is going to be step 1 that we'll go through, glycolysis, which is known as sugar breaking. Okay, why is it called sugar breaking? Because glucose will literally be split in half. Okay, so glucose will be split in this step. It's the initial breakdown to an intermediate. Okay, there will be some ATP produced in this step. A net gain of 2 ATP will be produced in glycolysis. Okay. Step 2 of this, I'm actually going to go down here and show you step 2. The name is right here. It's called oxidative respiration is the name of step 2. Okay, but it encompasses these 2 steps right in here, okay, which are the citric acid cycle, known as Krebs, because of the guy who discovered this. His name is Hans Krebs, also known as Krebs cycle, as well as the electron transport chain, which affectionately is abbreviated ETC. Okay, so step 1 in cellular respiration is going to be glycolysis, where glucose will be split, so breaking of glucose. We'll get a little bit of ATP. We'll get some electrons out of this. You have to always talk about the extraction of the electrons. Okay, and then step 2 is oxidative respiration. And if you see the word oxy, this is where the oxygen is going to be needed in this equation, or in this reaction. We're going to need it at step 2, which is oxidative. So one thing I want to point out to you is cellular respiration overall is aerobic, okay, A-E-R-O-B-I-C, aerobic, which means in the presence of oxygen. But don't be confused, because step 1 right here, glycolysis, is actually by itself anaerobic. Okay, anaerobic. Okay, anaerobic meaning that oxygen does not have to be there for glycolysis to take place, but this stuff down here, the oxidative respiration part, this is definitely aerobic. Okay, so when you put it all together, the entire process, cellular respiration, is known as an aerobic energy production pathway, but keep in mind there is a step of it, glycolysis, that does not have to have oxygen. This part down here, oxidative respiration, does need oxygen for it to occur, which includes Krebs cycle, and it includes ETC. Now, if you look at kind of these little mini steps in here about what's happening, Krebs cycle will complete the oxidation of glucose, okay, so it'll completely oxidize it. We will get our carbon dioxide as a waste product in Krebs cycle. It'll produce a little bit of ATP, but not much. Instead, more importantly, it's getting more electrons by breaking the sugars. Okay, the final step, the ETC, also known as oxidative phosphorylation, because we're dealing with phosphorylation, is our highest ATP yield out of the whole process, because energy will be released from a downhill cyclic flow of electrons in order to generate our overall ATP. Okay, so that's kind of the overview of cellular respiration. All right, hang on. Okay, so here's an overview slide of cell respiration. This is a really good slide to look at. I like this one. Hang on. Let's see. All right, so in this slide, overview of cell respiration, it starts off over here. All right, let's look at this overview slide. This is a great diagram of cell respiration. Okay, a lot of good stuff going on here. Let me move this over. Okay, so what you see is the picture of the entire cell. And so over here you've got the cytosol, okay, cell gel. And then over here you've got the mitochondria. Okay, so over here this is going to be step 1, number 1, called glycolysis, which occurs during the cytosol, and this is an anaerobic process. Okay, this is anaerobic. So you do not need oxygen present for this. In this step 1, you start off with glucose, C6H12O6, and we will end up splitting that, breaking it down during this step, which is called sugar splitting, until we get something called pyruvate or pyruvic acid. And these pyruvates are 3 carbon molecules, okay, 3 carbon, and you will get 2 of them because there's 1 glucose, which is a 6 carbon molecule. Okay, so go back and look at the big picture. Step 1, out here in the cytosol, which is anaerobic, no oxygen is needed, glucose, C6H12O6, will be split in half, because this step literally means lysis is to split, glyco, which is glucose. So you will split this in half into 2 3-carbon pyruvate or pyruvic acid molecules. Okay, now those pyruvic acids will make their way, as you can see with this arrow, into the mitochondria, where they will then go through both of these steps together are called oxidative respiration. Okay, so oxidative. I have a delay in my Moby pen, so that's why it's so slow. Oxidative respiration, which includes Krebs cycle and ETC, the electron transport chain. So both of those together are oxidative respiration. Okay, so back over here, step 1, glucose, C6H12O6, is going to be split into 2 3-carbon pyruvic acids that will then make their way into the mitochondria. Now there's some other stuff happening with this. Okay, one big thing, put a star by this, is when you split glucose, remember it's a 6-carbon molecule, which is covalently bonded together. If you split it, that means you're breaking a covalent bond. Well, when you break a bond, electrons are released. Okay, well, once again, we're going to have these great electron carriers. This time, because it's cell respiration, it's NAD+, not NADP, because NADP+ was for photosynthesis. Okay, this NAD+ picks up a couple of electrons with hydrogens and becomes reduced as NADH. Okay, so NADH is really important because it's been reduced, and its job, NADH, is to go up, as you can see this white line indicates, and over, and go way over until it gets until the ETC. Okay, down, oops, my arrow right here. And then once it gets to the ETC, those electrons are going to be dropped off and will go down little electron flow until finally we can generate the maximum amount of ATP possible, because that's the big thing that happens over here in the ETC is our maximum amount of ATP. Okay, so to get those electrons right here in step 1, we have to split sugar. When we split sugar, we're releasing electrons and they're picked up by NAD+. He gets over and goes to the ETC. Now, we also gain a little bit of ATP out of this. Our net yield is 2 ATP. It's a very small amount, and it's through substrate level phosphorylation. Okay, meaning using that enzyme, taking a phosphate, sticking it on an ADP, and putting those together to create that ATP. Okay, so that's step 1, and we're going to get into some specifics because there's actually an energy investment stage and an energy yield stage. We'll get into that in a minute, but I just wanted to kind of do the overview for you there. Okay, now move over to the yellow mitochondria. Over in the yellow mitochondria, you've got Krebs cycle, and then you've got the ETC, both of which collectively together make up oxidative respiration. Okay, now Krebs is actually in this space. Do you notice this area of the mitochondria? That's called the matrix of the mitochondria, M-A-T-R-I-X. Okay, that's in the matrix. It's in the, kind of like what I said, swimming pool area. If this mitochondria was a pool, it's in here on the inside. The ETC is going to happen on what's called the cristae, C-R-I-S-T-A-E, which is the membrane on the inside of the mitochondria. It would be like this line right here and coming around this line right here. So it's the cristae that fold back and forth. Like this would be a cristae right here, that thing. Okay, so Krebs is in the matrix and the space, and then ETC happens on the cristae. Well, what Krebs is going to do, because Krebs comes first, is the pyruvic acid that comes from glycolysis that enters, it's going to go through some changes where it will further be broken down, meaning we're going to take these 2,3 carbon molecules and break and break and break and break, and as we break, we get more electrons that are released. So once we release more electrons, those will be picked up again by NAD+ and FAD+, because there's another electron carrier called FAD. And so NAD+ and FAD+ pick those electrons up with the hydrogens and become NADH and FADH2. So these are reduced because they're carrying electrons. Okay, now where do they go? They're also going to go over to the ETC. So you can see the arrow showing them coming down as well. So those came from what was left over from our original sugar. Now the thing is, what's amazing is when you first split glucose in glycolysis, you're releasing less than 1/4 of the potential energy that's left in that sugar. And so that pyruvate has to go through some subsequent changes so we can continue to tap into that electron energy that's still stored in that molecule. And so that's why we go through those changes in Krebs cycle. Now, the cool thing about Krebs is one of our main waste products that comes out is carbon dioxide. Well, if you go back and look at your equation, carbon dioxide is a major waste product of cell respiration. It comes out right here in Krebs. Okay, now, do we also get a little bit of ATP? Yeah, once again, through substrate level phosphorylation, we'll gain a small amount. It's 2. So we gain a net of 2 from substrate level phosphorylation and glycolysis, a net of 2 from substrate level phosphorylation and Krebs, and then we're going to get our max, what I call our bang for our buck over here through oxidative phosphorylation, which is chemiosmosis. We're going to get a lot more over here. Okay, now at the end of Krebs, you are totally, like what you had from your original glucose molecule is completely broken down. There's nothing left after Krebs except for electron energy, which is in these carriers. So all the carriers, the NADH from glycolysis, the NADHs and the FADH2s from Krebs, they all collectively go to the ETC, where they will go through oxidative phosphorylation to make our major amount of ATP. Okay, so that's kind of the overview where you can see what's going on here. That is a really good slide for a review slide as well. All right, so we're going to get a little bit more specific with glycolysis. Okay, so glycolysis occurs once again. So, I mean, it's going to be probably remember all these things from me doing the overview slide, but I think it's important that we go through the specifics. So glycolysis is in the cytosol. Does not require oxygen, therefore we refer to it as anaerobic. Okay, make sure you know that. It's referred to as anaerobic. The first part of the pathway is an energy investment stage or phase, and then the second part is an energy payoff phase. So I will tell you, we are going to invest 2 ATP into this process. We're going to have to use, okay, that means use, 2 ATP in order to get an overall 4 ATP. Well, it's kind of like when you put money in the bank. If you put money in the bank, you start off with $1,000, you give the bank $1,000, and then you gain interest, and pretty soon you have like $1,010. You really gain $10. So in this case, we're going to invest 2 ATP, we're going to gain 4, so our net overall is going to be 2 ATP. Okay? So I'd like to explore this a little bit closely, a little closer by showing you what's going on in this slide. So hang on, let's get, take a short break. Okay, so here we go. Let's talk about the energy investment part of glycolysis. See if I can move this down a little bit for you. All right, so the first thing, there are going to be many enzymes used in this process. The first one you see listed here is hexokinase. Okay, so hexokinase will first transfer a phosphate group from ATP, in other words, break off this P, okay, what you're left with is ADP. That phosphate is then going to be transferred, okay, to glucose right here. You can see it's added on, transferred to glucose, making it more chemically reactive. The charge on that phosphate also helps trap the sugar and keep it in the cell. Okay, so that's the first thing that's going to happen. Then what we have now is called glucose 6-phosphate, because now it has an extra little phosphate there. It's converted to its isomer form called fructose 6-phosphate. So it goes through a small change to become converted into an isomer form, which you can see that the chemical change of the shape of it has changed a little bit. So now it's in its isomer form called fructose 6-phosphate. Okay, then phosphofructokinase, another enzyme, see it ends in ASE, okay, so another enzyme comes in and transfers another phosphate from another ACP, okay, transfers that to the opposite end of the sugar. And I don't know if you can see in my picture. Let's see if I can scroll down a little bit. Wait. Yeah, it's over here. You can see it on this end. Transfers another phosphate to the opposite end of the sugar, investing a second molecule of ATP. Okay, so that's using 1,2 ATP. Okay, this is a key step for the regulation of glycolysis. Okay, then after that, another enzyme called aldolase, see it listed right here, cleaves, which means breaks the sugar molecule into 2 different 3-carbon sugars. Okay, these are isomers of each other. Okay, so one of these is called dihydroxyacetone phosphate, and one is glyceraldehyde 3-phosphate. So that's the first step, which is the energy investment phase. Okay, then the payoff phase is next, where we're going to actually create some ATP in the second one. All right, in this part, first thing I want you to notice is the little subscript 2 that you see everywhere in this diagram. The reason that the 2 exists there is because in that energy investment phase, we ended up with those 2 3-carbon compounds. Okay, so because there's 2 of them, the coefficient 2 is going to precede all molecules in this phase. I just wanted you to understand that that's where that was coming from. Okay, what you have going on in the energy payoff phase, first off, there's an enzyme. Okay, you can see the dehydrogenase used here. There's going to be an enzyme that catalyzes 2 sequential reactions back to back. First, the sugar is going to be oxidized. Okay, how is it going to be oxidized? By the transfer of electrons to NAD+, forming our NADH. Okay, so we're going to oxidize the sugar by transferring electrons, so the NAD+ is going to be reduced. Okay, second, the energy that's released from this exergonic redox reaction is used to attach a phosphate group to the oxidized substrate, making a product of very high potential energy. Okay, so you can see the phosphate group that's used in this one to attach the phosphate group to the oxidized substrate. Okay, so you see your phosphate added on right here. Okay, next, the phosphate group that was added in that step is transferred to an ADP, okay, through substrate level phosphorylation in an exergonic reaction. Okay, the carbonyl group of the sugar is then oxidized to the carboxyl group in this case. Okay, another enzyme you can see listed here, phosphogluceromutase, relocates the remaining phosphate group. You see the relocation from here to this. Okay, then there's another enzyme, an enolase. And by the way, you do not have to memorize all of these enzymes. I'm just like showing you a big picture overview of what's happening in this energy payoff phase. Basically, you have some rearrangement happening, causing double bonds to occur, removal of water. You've got all this stuff happening, and then over here at the end, you have another formation of ATP through substrate level phosphorylation. Okay, so just don't worry about memorizing all of these compounds. You don't have to, memorize those and know the names of these enzymes. The big picture is, if you go back to this one, we are investing a little bit of ATP. How many we're investing to? Why? Know kind of what's going on in that to invest it. Where does the investment come from? Okay, what happens when you break a P off? What does that P do? It's added to these. So you're kind of knowing big picture. And in this one, same thing. You're taking the sugar, the 2 sugars that you started off with, and you're doing some changes to them along the way. As you're changing them, the changes are occurring because you take some phosphates in some enzymes along the way, and you are generating some ATP. Okay, so it's 2 ATP here, 2 ATP here, over here, and 2 different parts of it. Where do the 2's come from? Because you have 2 sugars going through this. So each sugar is generating 1 and 1, 1 and 1, and that's where you get your overall 4 ATP along the way. So if I invest 2 and I get 4, that's where my net yield of 2 ATP from glycolysis comes from. Okay, and then in this slide, you can kind of see the overview, same thing. The summary of glycolysis, where you start off with sugar, glucose, and you go through a little change where you split that in 2. Okay, so that's the energy investment phase. You use 2. What do you get from that? Well, left over. If I'm using 2 ATP, I break a P off, I get ADP, and I get inorganic phosphates. That then enters into the energy payoff phase, where I'm forming 4 ATP. Okay, why? Because I'm sticking P's onto ADPs through substrate level phosphorylation. Okay, what else do I get? I get these electron carriers. Okay, these are the NADHs. Those are going to go to the electron transport chain. So each of these electron carriers are carrying high energy electrons. They've been reduced. Okay, and you can see over here they were NAD+, plus we added those electrons and we added those hydrogens, and now we get an overall 2 of these NADH. Okay, as a result, I now have compounds called pyruvate or pyruvic acid, and of course, I formed a little bit of water in this process. Okay, so the net at the bottom, you can see glucose becomes pyruvic acid or pyruvate. My 4 ATP formed minus my 2 U's gets me 2 overall net gain, and then my NADs that caught the electrons along with the hydrogens became my 2 NADHs, plus a little bit of hydrogen as a result of that. And those hydrogens lead to the buildup of a gradient that will help with chemiosmosis later on. Okay, so that's glycolysis. All right, moving on to now, let's go to oxidative respiration. We're going to take the 2 pyruvic acids that were formed in glycolysis, and we're going to do more to them. Why? Because they still contain a tremendous amount of chemical energy. They say that less than a quarter of the energy in one glucose is harvested in glycolysis. So we still have a tremendous amount of energy left in that glucose that we have to do something with. So if oxygen is available, and you might say, well, Ms. Lynham, when would oxygen not be available? If you were in a condition where you were exercising excessively and you were going through oxygen debt, then you might not have enough oxygen available in the cell. And so you might not be able to do this oxidative process if you were in an exercising excessive condition. But if oxygen is available, that pyruvic acid is going to enter the mitochondria and go through citric acid cycle, or Krebs, and further oxidation. Okay, but before we can get to those 2, there is something called a grooming step. Okay, so upon entering the mitochondria, right as it gets inside, but prior to going into the citric acid cycle, we're going to do something called grooming. Okay, it's a grooming step. And that's where we're going to change these pyruvic acids a little bit by grooming them. Okay, so in the grooming step, this is actually really critical because it's prior to the citric acid cycle. We're going to take the pyruvic acid. The first thing that's going to happen is a carboxyl group, remember it's a functional group, which is present on pyruvate, is going to be removed. Once it's removed, it will be given off as our first carbon dioxide waste product. So CO2 is going to first be removed in this grooming step. Okay, the remaining 2 carbon compound is going to be further oxidized, remember oxidized by extracting electrons, to something called acetate. So if we oxidize it by removing those electrons, the electrons have to go somewhere. They're going to be given to an NAD+. So we're going to reduce NAD+ to NADH plus some hydrogen. Okay, that remaining acetate is going to bind quickly to a molecule known as a coenzyme A. So once it does that and it binds to it, it becomes something else, and our product at this point becomes acetyl-CoA. So big picture for you looking at this grooming step, guys, you need to know basically that in the grooming step, we're going to take pyruvic acid and we're going to groom it into acetyl-CoA. Okay, so we're going to groom pyruvic acid into acetyl-CoA. How do we groom it? We break off something. We're breaking off a carbon, breaking that off, and then we have carbon dioxide as a waste. We get a little bit of electrons, and then this all happens because it's reacting with a coenzyme A to form our acetyl-CoA. So let's look a little bit closer at this. Here's a great picture. I really like this one, so let me see if I can get my thing up. Okay, so here's your pyruvate at the bottom, okay, also known as pyruvic acid. It is a 3-carbon compound. You see the 3 carbons? And it has a very reactive carboxyl group right here on the end. Oh, this Moby is driving me crazy. Okay, very reactive carboxyl group. Okay, and it is so reactive, it's so charged, I just wanted you to know that the only way for it to get into the mitochondria is through this transport protein, and that is a type of active transport. So it has to get in and go from low to high across that mitochondrial membrane to get in. Okay, so once it gets in, okay, first thing that's going to happen, number 1, is carboxyl group is cleaved off. That's where you get this carbon dioxide from. So that's the cleaving of the carboxyl group. Okay, as that's cleaved off, okay, the oxidation of that is going to leave some electrons that are released that are caught by NAD+ to become NADH. So that's the oxidation of that pyruvate once it enters into the mitochondria. Okay, after that, third thing is you've got something called coenzyme A in here in the mitochondria that reacts with what was left of the pyruvate, and now we have something new called acetyl-CoA. So we can't call it pyruvic acid anymore because it's only a 2-carbon molecule. Do you guys see how it's a 2-carbon? Okay, 2-carbon molecule, 1, 2, because one of those carbons left as this waste product over here as carbon dioxide. Okay, so this is called the grooming step. Make sure you know this. It's right before we go into Krebs cycle. We take pyruvic acid. We first cleave off the carboxyl group. When we do that, that becomes the carbon source for our carbon dioxide. We get electrons, which are passed to NAD+. They become NADH. Those guys will go over and go make their way to the ETC is where they're going to go. Coenzyme reacts with what's left of pyruvate to become our new molecule called acetyl-CoA, which is a 2-carbon compound. All right, so moving on to Krebs cycle, also known as citric acid cycle, tricarboxylic acid cycle, you name it, it's got different names. But just as the name implies, it is a cycle. What does a cycle mean? You're always going to have something spinning in the cycle that will always be present. So if you look in this diagram, up here at the top, here's the grooming phase that we just did. Okay, so from up at the top right here, pyruvate, to get to acetyl-CoA, that's the grooming that occurs. So now you've got acetyl-CoA coming into the cycle. Okay, CoA, by the way, because it's a coenzyme, just remember it's always there, it's always recycled. But when acetyl-CoA comes in, it's going to go through a series of redox reactions where it will turn. Once again, this is happening in the matrix. What we're going to get from this big picture is some NADHs, we're going to get some FADH2s, we will get a little bit of ATP, but more importantly, we are going to completely break down this acetyl-CoA, so we have nothing left of our sugar by the time this Krebs cycle is done. So we're going to produce quite a bit of carbon dioxide as a waste product in the Krebs cycle. Okay, now let's look more specifically. Okay, here's the Krebs cycle. Okay, so looking more specifically at Krebs cycle, here's your acetyl-CoA coming in, okay, from our grooming phase. So here it is coming in. Already in the cycle, spinning over here is this big dude, I like to call him Big O. Okay, it's oxaloacetate. It's a 4-carbon compound that's in the cycle. So this 4-carbon compound comes around, joins with a 2-carbon acetyl-CoA. So they briefly join together and become a 6-carbon compound right here called citrate, which is why this cycle is called the citric acid cycle, because citrate is citric acid. Okay, so that's the first thing that's formed by the joining of acetyl-CoA and oxaloacetate forms the citrate. Okay, that citrate then is going to go through a little series of changes from here to here to become isocitrate. How does that happen? It's a little bit of isomerization. It's converted using some water. Okay, so it's converted to that. Then that isocitrate between here and here is going to be oxidized. Okay, so some changes are going to happen. We're going to break a bond. As we break a bond, we're going to release a carbon dioxide molecule. Okay, so the electrons have to go somewhere, so waiting right here is an NAD+ to receive the electrons to become NADH. Okay, so the resulting compound loses that carbon dioxide. Okay, then another carbon dioxide is going to be lost again, as the next one is oxidized as well, doing the same thing. NAD+ receives some electrons, we release some CO2. the remaining molecule is then going to be attached. You'll notice another CoA down here. It's attached to coenzyme A by an unstable bond. Okay, so that's coming around at this point. Now, down here at the bottom, CoA is displaced by a phosphate group, which is transferred to GDP, forming GTP. It's a molecule that functions similar to ATP. It's also used to generate ATP down here. So it's just a kind of an intermediate form that helps in the production of a little bit of ATP. So we're changing this along the way. CoA then comes out and can be recycled. 2 hydrogens are transferred to FAD, forming FADH2. Okay, so that probably is like right here. So we get a little bit of FADH2. And now we have oxidized something called succinate. Okay, at that point, we have some water come in that will do some changes along the way. Succinate becomes fumarate, okay, rearranging the bonds. That then is rearranged a little bit more. A few more changes, becomes something called malate, which then becomes changed, a little bit of changing going on as well, another breaking of a bond, receives some electrons, and we get big O, who is in the cycle and stays in there and is ready to receive another acetyl-CoA. So this Krebs cycle, like I said, don't worry about memorizing steps so much as you're memorizing big picture of what's going on. We're starting off with a 2-carbon compound coming in, which originally came from the glucose that started off this process. We're joining it briefly. Why are we joining it briefly? So we can release the remaining carbon dioxide, which is our waste product of cell respiration. Okay, as we do that, then we add another coenzyme, do some changes down here at the bottom, generate a little bit of ATP, but then we're left with something in the cycle called oxaloacetate that's waiting to do this whole thing again. So in the matrix of the mitochondria, you must have adequate enzymes, you must have NAD pluses, you must have this oxaloacetate waiting to keep doing, waiting to do this to this acetyl-CoA that comes in. Okay, so that's kind of what's going on with Krebs cycle. Okay, now moving on to the final step of oxidative respiration, which is oxidative phosphorylation, traditionally called the electron transport chain. Now most colleges and textbooks will just generally call this oxidative phosphorylation, but it is the ETC. It occurs in the inner mitochondrial membrane, which is called the cristae. The membrane is organized to have a lot of cristae. You can see in this diagram, look at the folds back and forth and back and forth and back and forth. The more highly convoluted these folds are, the more surface area which is provided for the ETC to function. Okay, so there's 2 important components of this oxidative phosphorylation that we're going to go through. We've got electron transport, and then of course we have the formation of ATP through chemiosmosis, which is why this step is so important. Okay, so here we go. Okay, so the electron transport chain literally is, this is what it is, it's a collection of molecules. You can see in the picture here, all these like FMN, FES, Q, all these listed along the way. It's a collection of molecules, each more electronegative than the one before it. Okay, and the way that it functions is you've got all these NADHs and FADH2s, which came from glycolysis and Krebs cycle. So they're going to come to the ETC, they're going to drop off their electrons. So the electron transport chain proteins, these guys, the purple ones, are going to be reduced as they receive electrons. Then they're going to be oxidized as they pass the electron down the chain to the next one. Okay, so they're going to first receive the electron from NADH and FADH2, which will reduce them briefly. They'll pass the electron on, and then they'll go back to being oxidized as they pass that electron on. Okay, at the end of the ETC, the ultimate electron acceptor, you can see down here in red, is oxygen. That's why oxygen must be needed for this process, y'all. It's oxidative respiration because it is the final electron acceptor at the end of the ETC. Now the purpose of this, the whole ETC purpose, is to establish a hydrogen ion gradient on 2 sides of the inner mitochondrial membrane. So we want to establish a gradient or a buildup of hydrogen ions. Okay, the energy from these falling electrons is going to be used at the very end to pump hydrogen ions from the matrix into the inner membrane space, which will ultimately make our ATP. Okay, so chemiosmosis is one of the steps at the end, and that's going to be the making of the ATP. Okay, first of all, we have enzymes that are used here, ATP synthases. Look at the name, synthase. ACE means enzyme, synth means to make. What are we making? ATP. And when I say make, we're not really making ATP. We're taking a phosphate, an inorganic phosphate, and we're sticking it onto an ADP, which is already present. So down here in the picture, you can see there's your ADP, there's your inorganic phosphate. So for us to quote-unquote make it, we're sticking the P on the ADP to create over here ATP, okay? Now, ATP synthase in this diagram is this purple thing. It's a really unique kind of protein enzyme that is functioning by spinning. It's like a rotor, okay? So it's constantly spinning in order to do its job. It is located in the inner mitochondrial membrane, okay? So here's the membrane. You can see this phospholipid bilayer because that's what the membrane is. Now, y'all, one thing I want to point out here, remember, when this happens in a prokaryote, a bacteria, they can still go through oxidative respiration, but they don't have a mitochondrial membrane because they don't have membrane-bound organelles. So this would occur in their plasma membrane or cell membrane. Okay, so they can still do this, but it's just in a different location. Okay, so these enzyme complexes, like this purple one called ATP synthase, located all along the mitochondrial membrane, is going to help. What will happen is we will have this buildup of hydrogen ions. You can see there's a gradient over here on one side. This is also known as the proton motive force, kind of like what we studied in the transport section. So the movement of these H's through this ATP synthase, okay, rotates the enzyme, makes the enzyme spin. As it spins, the rotation, it exposes the active sites that are in here, because this is an enzyme, y'all, so the enzyme has an active site. So as it spins, the active sites become open, which then can fit your ADP and your P. Those can join together to form our ATP. Okay, here's an overview of the electron transport chain. So you can kind of see everything together all in one diagram at the ETC. So it's broken down, you can see down here at the bottom, into the ETC. Okay, see the little bracket, electron transport chain? And then down here, this is really chemiosmosis, which is, it says oxidative phosphorylation, also known as chemiosmosis. So what you have, okay, let's just do the big picture here, is we are trying to make ATP. That's the overall goal. So to do that, all we have left coming from Krebs cycle or citric acid cycle and coming from glycolysis is right here. You can see I'm circling it with my thing. We have NADHs and we have FADH2s coming here. So the goal is we got to get these NADHs and FADH2s break off or drop off the electrons. Okay, so they're going to drop it off. Now follow the arrow right here in gold or yellow. This is the pathway of the electron traveling. Okay, it's going to travel across, these are complexes. Okay, all these purple molecules, they're proteins that are embedded in the mitochondrial membrane, and these are different complexes. And as the electron, what's in gold, passes through these, remember, there's a lot of energy in there, these things are going to be reduced. Well, as that happens, hydrogens are going to be pumped through, okay, you can see the H is pumped through, they're going to go through these proteins and contribute to the buildup of a gradient of hydrogens on one side of this membrane, and this is actually the inner membrane space. So NADH and FADH2 break off electrons. The electrons pass through from complex to complex to complex. I do not want this. Pass through from complex to complex, and as they pass through, hydrogens are pumped to the other side, okay, and build up a hydrogen gradient. Once the electrons get down to the end, look at my cursor where I'm at, they get way down here at the end, what's waiting is oxygen. Oxygen is the final electron acceptor. So the oxygen receives the electrons, plus receives some of the hydrogens that are over here. How are they over here? Because we already have some of the H's that come off, and some are pumped through, but some stay on the side. And then they add to that to create water. That's our waste product of this. Okay, so that's the flow of electrons, is from complex, 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 received by oxygen to form our water. Okay, now the second part of this is this buildup of hydrogens on one side, on this intermembrane space. These are then going to go down the gradient through the special ATP synthase, which is the special protein molecule. When they're pumped through, okay, they're coming through down their gradient, that force is what opens up the active sites on this enzyme, because like I said, this is an enzyme that has active sites. The active sites are exposed, and this thing has like a proton motive force. It spins and grabs the P's, sticks it on ADPs to create ATPs, and that's our oxidative phosphorylation. Okay, so you got kind of a lot of stuff happening here, but that's to create the maximum amount of ATP possible. Okay, looking at, once again, here's overall summary. You can see what's happening, everything in one picture. Glycolysis is after in the cytosol. We get 2 ATP. Okay, we get 2 NADH that travel over and are going to make their way to the ETC. Okay, then the pyruvic acid, which is attained from glycolysis, enters into the mitochondria. It first goes through this pink is a grooming phase. There's a grooming phase. It's the conversion of pyruvate into acetyl-CoA. We still gain a little bit more NADH. We also, out of this, got a little bit of carbon dioxide. See, that's not shown on this diagram, but we get a little CO2 right here. Okay, the acetyl-CoA goes in through the Krebs cycle. Okay, goes around the cycle, joins in with big oxyl acetate. It's changed. We eventually get more CO2 come out from this. We gain 2 ATP, but more importantly, we get NADH and FADH2, which also shuttle over to the electron transport chain. Okay, step 3 over here at the electron transport chain, we then go through oxidative phosphorylation and chemiosmosis, where we'll take all the FADH2s, the NADHs from along the way. They will dump off their electrons, which will go through the electron transport chain, and we will gain between 26 and 28 ATP. Okay, so the maximum amount per glucose, if you add all this up, is between 30 and 32 ATP is what our total is. All right. Hang on a sec. Okay, now one thing I wanted to mention here on this slide is there's been a debate as to exactly how many ATP can be generated for one glucose molecule. It's still one of those things that they're debating even now. A lot of it has to do with, I think there's 3 different mechanisms that they look at. One is there's still some debate as to when the hydrogens are pumped through an ATP synthase. There's a lot of debate about how many hydrogen ions it takes to go through that to generate 1 ATP. Some scientists were first saying that it takes 10, and then they're going back and forth, and the new number that they agreed upon was they said, oh, it takes 4 hydrogens to be pumped through an ATP synthase to generate an ATP. So it's kind of debatable on that. So what the new, I guess, 2013, the new things that they're releasing that we're going to go with is that for each NADH, this little bullet point down here, each NADH that's shuttled through the ETC, the yield is about 2.5 ATP. For each FADH2, the yield is about 1.5. So if you go up here to the diagram, okay, and this is another contention about why it's not sure about how many ATP, as you can see up here, there's a little split after glycolysis. There's some debate on how the shuttling takes place of these NADHs. Some think that it's NADH that carries it. Some think that there's a slight change and it's FADH2. So depending on which one of these two carries the electrons over to the ETC after glycolysis, that's going to give us a different number down here as an end result. So there is some debate. So what we're going to go with for my class is we're going to say that each NADH supplies 2.5 ATP, each FADH2 supplies 1.5. So if you go along and add them up, let's just say we start with this 2 NADH is really this one. So if we have 2 here, that's 4, plus 6 here, that's 10. We're going to have 10 that are supplying us with 2.5 ATP each. So it's not even a whole number. So you do the math with that. And then the FADH, we're not going to use this one because we went with the NADH route. So all we have are these 2 FADH2s, and each of those is 1.5 ATP. So if you multiply those out, I think that's where this number 28 ATP comes from. So that 28 ATP was generated through chemiosmosis and oxidative phosphorylation. So 28 plus 2 here is 30, plus 2 here is 32. So depending on if you use that route or if you go back up here and you say, oh, let's not use NADH, let's use FADH2, and so these 2 up here would be 1.5, that's where you get that 26 from. So between 30 and 32 total is what is acceptable on this. All right, last, not last, but next little slide is talking about cellular respiration of things other than glucose molecules. So if we use other foods besides glucose, let's say we use proteins or we use fats to be able to run this process. Let's look at what goes on with this. Okay, it's important to talk about the cellular respiration of other molecules because as humans, our diet does not just consist of glucose. Most of our calories come in the form of fats, proteins, sucrose, and other disaccharides, and starch, which is a common polysaccharide. So it's good that you know that this can be used by, we can use this for many different food molecules. Okay, so look at the first little purple column right here, which shows proteins. So let's say we eat proteins. First off, we're going to break them down, digest them into their monomer form of amino acids. Okay, at that point, though, what we have to do before we can take the product and enter into glycolysis, or take the product and enter into the acetyl-CoA formation, the grooming step, or even into Krebs cycle, we have to do something called deamination. And deamination is the removal of the amino group. When we remove this, the amino group becomes part of our waste. We excrete it in the form of urea in our urine, and then what we have left are the products that can enter into some part of cellular respiration along the way. Up here in the middle section, you see carbohydrates. These sugars that enter into glycolysis, this could be glucose, fructose, galactose, but let's say it's something like glycogen that we convert into glucose along the way. We'll be digested and breaking it down, and then those can eventually enter into glycolysis. Okay, in the yellow part over here, we can also use fats. Remember, fats can be broken down into glycerols and fatty acids. Glycerol can be immediately, once we break it down, converted into a 3-carbon molecule called glyceraldehyde 3-phosphate, which can plug right into glycolysis, be converted into pyruvate, and then continue the same cycle of going through cell respiration. Fatty acids, we can oxidize those, break those down into 2-carbon compounds, which enter right at the acetyl-CoA portion of the grooming step before Krebs cycle. So we can use more than just glucose for cellular respiration. I just wanted to make sure I mentioned that. Okay, the last slide for cellular respiration is talking about some metabolic poisons. I thought this was important to mention that there are some poisons, I'm sure you've heard of cyanide or carbon monoxide, which you can see in the middle of this picture, which the direct effect is blocking the ETC. So it's the inhibition of ATP synthase that can cause a major problem. Ultimate effect, what do we have, is decreased ATP production. If you have decreased ATP production, then symptoms might be lethargy, low energy levels, not being able to perform to max capacity because you're not having as much ATP. These metabolic poisons are known as uncouplers because they're preventing the creation of a hydrogen ion gradient because there's a leakiness of the mitochondrial membrane. So instead of these hydrogens coming through like they're supposed to come through and be pumped to the other side, you can see in this one that if you've got this poison that's blocking the flow of hydrogen, the hydrogen stays on this side of the membrane. So if we can't build up that gradient, then that's going to affect the last step down here, which is chemiosmosis, which is where we're supposed to be having the hydrogens come through down their gradient. Well, if there's no big gradient that was established, then this last step is going to have a huge major impact. So you'll have decreased ATP production as a result of that. Okay, that's it for this video. Look for the next one. I'm going to post one on fermentation.